Dear students, today we are going to see about the microanatomy of GIT, mainly two slides, esophagus and stomach. Under the stomach we have two slides, fundus and pylorus. So totally we are going to see about three slides. Before that, you should know about the common layers of GIT. It is common for all the parts of GIT. So your esophagus, stomach, small intestine, large intestine, you will be having these common layers. Inner to outer, first one is mucosa. The mucosa composed of three components. So that is epithelium. Epithelium supported by a layer of connective tissue called as lamina propria, then muscularis mucosa. So these three combined together, it is called as mucosa. So you should know the difference between epithelium and mucosa. Mucosa means epithelium, lamina propria and muscularis mucosa. So next layer outer to the mucosa, what we have is the submucosa. Then still outer to that you will be having muscularis externa. We used to call it as muscular layer. It is made up of two layers. One is inner circular layer and outer longitudinal layer. So it is made up of smooth muscle fibers. Next outermost is called as serosa or adventitia. Serosa means if that abdominal organs, for example, stomach, intestine, colon, and all, covered by peritoneum, so that covering is called as serosa. Otherwise, you will be having a thin layer of connective tissue that will be called as adventitia. So these are the layers of your GIT, gastrointestinal tract. See this picture. So this is the lining epithelium supported by a layer of connective tissue called as lamina propria. So next outer to that you have a thin layer of smooth muscle fibers called as muscularis mucosa. So mucosa means, so in the exam the examiner may ask what are the components of mucosa. So what you have to answer is epithelium supported by the connective tissue called as lamina propria, then muscularis mucosa. Combined together it is called as mucosa. So that is the inner lining. Next outer to that we have another layer that is called as submucosa. So submucosa, it is made up of moderately dense connective tissue with blood vessels and nerves. So that we will see one by one in the other slides. So next outer to that you will be having muscular layer. It is called as muscularis externa. So that is made up of two layers, inner circular layer and outer longitudinal layer. Still outer to that we have serosa. So these are the four common layers for all the GIT organs. Your esophagus, stomach, small intestine and large intestine you are going to see. So first identification point for GIT slides, presence of these four layers. So next we will see about the esophagus. So that is the first GIT slide we are going to see. So the sites where friction, wear and tear are more common means what will be the lining epithelium? So already you studied about the epithelium. So if there is more friction, wear and tear at that site, you will be having epithelium called as stratified epithelium. So stratified means multiple layers of cells will be there. That only if you take your esophagus, the lining epithelium of esophagus is your non-keratinized stratified squamous epithelium. Non-keratinized stratified squamous epithelium. Already we studied about that stratified squamous epithelium. It is having two types, keratinized and non-keratinized. So here what we have is non-keratinized stratified squamous epithelium, important for MCQ. So here clearly you are able to make out that mucosal folds. So that epithelium is your non-keratinized stratified squamous epithelium. You are able to make out numerous layers of cells. So that means it is stratified. So it is stratified squamous epithelium non-keratinized type. So it is supported by a layer of connective tissue, that only already I told, it is called as lamina propria. It is made up of loose areolar tissue or loose connective tissue. It is having your lymphocytes. So here you are able to make out aggregation of lymphatic nodule also, but you will be having lymphocytes, blood vessels you can make out in the lamina propria. Then already I told mucosa means you will be having a layer of muscularis mucosa, that only here you are able to make out. This is a longitudinal layer of smooth muscle supporting the your lamina propria. So this is your muscularis mucosa. So that is here it is longitudinally placed. So that's why you are able to make out like this. So now this is the mucosa of the esophagus. So already I told mucosa means lining epithelium, so supported by connective tissue and a layer of smooth muscle called as muscularis mucosa. In case of esophagus, it is longitudinally placed. So now this is the mucosa of your esophagus. So easy to identify. So if any luminal structure with stratified squamous non-carcinate epithelium, next you have to see for submucosa. So this is our submucosa. So up to this it is mucosa. Now this part is the submucosa. As I told already, it is made up of moderately dense connective tissue with elastic fibers, collagen fibers, blood vessels. You will be having blood vessels and nerve plexus also. So here the identification point for esophagus is you are able to make out group of mucus acini. So these are all mucus acini. So you are very clearly you are able to make out group of mucus acini. So this is the identification point for esophagus. So the lining epithelium is non-keratinized stratified squamous epithelium means C for the submucosa, where in the submucosa you are able to make out that mucus secreting acini means that is your esophagus. So clearly in this picture you are able to make out one duct of this mucus acini opening into the surface epithelium. So why we have that mucus secreting acini in the submucosa? It is for lubrication. So if you take any food, so that easily it should go and reach the stomach. So for the lubrication of the food substance, we have that mucus secreting gland. Okay, so why we have non-keratinized stratified squamous epithelium? So as I told already, wherever there is more friction or wear and tear, that's in at that site, you will be having stratified epithelium. So multiple layers of cells you will be having. So here if there is any tear, if we will taking hard substance or any hot substance also, there will be epithelial cells injury. So automatically that will be replaced by regeneration of the basal cells. So that's why we have stratified squamous non-keratinized epithelium here. So this question may be asked, what is the function of stratified squamous epithelium in esophagus? Next question, what is the function of mucus secreting glands present in the submucosa? So that these 2 points are important for the identification. So you are getting this slide means 2 identification points you have to write for spotters. One is presence of lumen. So next is presence of all the 4 layers of GIT. So next lining epithelium. So that is your stratified squamous non-keratinized epithelium. Next, presence of mucus acini in the submucosa. So these are all the identification points you should be able to tell. Next, if you come out, so mucosa, submucosa, then your muscularis externa. So this is muscularis externa, mainly made up of inner circular. So this is inner circular smooth muscles. Next, outer to that you will be having outer longitudinal muscle layer. So this is uniform, inner circular and outer longitudinal. But regarding muscle coat in esophagus, we will be having some difference. So if you take your upper 1/3, where as your esophagus is continuation of your pharynx, you will be having continuation of the skeletal muscle fibers of pharynx into the upper one third of the esophagus. Next, if you come down to the middle third of esophagus, where you will be having mixing of both smooth muscles and skeletal muscles. If you take at the lower end of your esophagus, where you will be having entirely smooth muscle fibers. So this may be asked in the viva. So now outer layer is your serous layer. So here in case of esophagus at the upper end all, you will be having only adventitia, that is a loose layer of connective tissue with blood vessels and nerves. So if you take that lowermost end of your esophagus, where you will be having your serosa. So now these are all the features of your esophagus. So with these points only, you have to see this slide. First see for the lumen, then see for the epithelium. So if it is non-keratinized stratified squamous epithelium, next you see for the submucosa. The submucosa, if you are able to make out that group of mucosacini, then it will be esophagus. So these are all the points you have to remember regarding your esophagus. Okay, so next we will see about the stomach. Under the stomach, as I told, we have two slides. One is stomach fundus, other one is stomach pylorus. So first we will see the slide of stomach fundus. So first what you have to note in this stomach is presence of that four layers. So this is your mucosa. So this is your submucosa. Next you will be having your muscularis externa. Next this is our serous covering. In case of stomach, it is lined by peritoneum. So you will be having only serosa. So this is serosa. So we have four layers. So mucosa, submucosa, your muscularis externa and serosa. So basic four layers are there. So it is one of the slide of GIT. So adjacent to that, you are able to make out another image. So it is nothing but this is low power, this is your high power. Next, you have to see the mucosa. So mucosa means, as I told already, epithelium, lamina propria, and your muscularis mucosa. So composed of your mucosa. So in the esophagus slide, we have seen the lining epithelium is non-keratinized stratified squamous epithelium. So if you trace it down, so your esophagus only continues as stomach. So at the esophagus, at the junction of esophagus and stomach, you will be having change of that epithelium. So if you take your stomach, here the main function of stomach is secretion. So it is made up of simple columnar epithelium. So that will you have to see here. So this is high power. You are able to make out that simple columnar epithelium. So stomach is lined by simple columnar epithelium. So that is the lining epithelium. So here the main function is secretion. So you will be having secretion of mucus, very thick mucus. So that will cover the entire inner surface of the stomach. As already you know that you studied in physiology. So we have parietal cells in the stomachs that secretes HCl. So to protect that, you will be having thick mucus secretion from the cells. So next important point what you have to see in mucosa is, so if you see that epithelium that is dipping into the lamina propria. So that epithelium is dipping into the lamina propria. The site of dipping of the epithelium into the lamina propria is called as gastric pit. So see that. So these are all the gastric pit where your epithelium is dipping into the lamina propria. So what happens to that epithelium after that? After dipping into that lamina propria, they are getting converted into glands. So now what you are seeing here are the glands present in the fundus. So these are all the glands present in the fundus. So these glands are called as fundic glands. Okay, so now what are all the cells we have there in the fundic glands? So that is important. So already you have studied in physiology. So you will be having three types of cells in the fundus glands. So First important one is your parietal cells. So here you are able to make out. So this is high power. So beaded appearance you are able to make out here. So that beaded appearance is due to presence of parietal cells. So what is the other name to parietal cells? Your actinic cells. Already you studied in physiology. So what is the main function of these parietal cells? So that secretes your HCl. Okay, so that is the identification point for your fundus. So you are able to make out that epithelium, gastric pit. Deep to the gastric pit, you are able to make out that gastric glands. Okay, so these are all the glands. In the glands, what are all the types of cells you have? So if you see the upper part of the gland where you have parietal cells. So all these parietal cells, otherwise called as actinic cells, are very important. So they are stained with eosin. Clearly you are able to make out. So they are all pink cells. They are very close to the basement membrane that gives the beaded appearance. So that is the identification point for your fundus. Next, if you go down to the lower part of that gland where you have hematoxylin stained cells, so these cells are called as chief cells, or it is called as zymogenic cells. So that also already you studied in physiology. So it secretes pepsinogen, so it will be getting converted into pepsin. So in the lower part of the gland you have hematoxylin stained cells. They are the chief cells. So in the upper part of the gland you are having more of parietal cells. One more type of cell present very close to the gastric pit are called as mucous neck cells. Mucous neck cells. Actually that beginning part of the gland is called as isthmus, where you will be having the same mucous cells what we have in the surface epithelium. So they are called as mucous neck cells. So that will secrete once again the mucus. So these parietal cells secretes your hydrochloric acid. Any other important secretion by your parietal cells as you studied already in physiology? Yes, very good. Your intrinsic factor. So that is very important. Intrinsic factor is secreted by your parietal cell. So that is more important for the absorption of vitamin B12. So if there is any defect that leads to pernicious anemia. So that already you have studied in physiology. So now if you come down, you are able to make out your muscularis mucosa. So now this entire part of that slide is mucosa only. Note that up to this. So now this is the surface epithelium. So now this is the gastric pit. Already I told what is it. The site of dipping of the epithelium into the lamina propria is called as gastric pit. Beyond that, what we have is the gastric gland or fundic gland. Okay, so there mainly we have three types of cells: mucous neck cells secreting mucus. Next, your parietal cells or actinic cells secretes your hydrochloric acid as well as intrinsic factor. Next, if you go down deep to that, you will be having your once again chief cells or your zymogenic cells secreting your pepsinogen. So the function and all already you studied in physiology. Now this is the your muscularis mucosa. See the difference between the muscularis mucosa of esophagus and this. So here you are able to make out inner circular layer and outer longitudinal layer of your muscularis mucosa. So now entire thing is your mucosa. Now if you go to the submucosa, so this is your submucosa. So as I told already, the common features, it is made up of moderately dense connective tissue. So you will be having blood vessels, nerves and all you can make out here. Your lymphocytes, diffusely placed lymphocytes and all you can make out. So that is common for your esophagus also. So next important point, in that submucosa you will be having plexus of nerves. What is the name of that nerve plexus present in submucosa? it is called as Meissner's plexus, Meissner's nerve plexus. Okay, so next outer coat, if you go, you will be having your muscularis externa. So that is circular. So this is inner circular and outer longitudinal layer. Now in between the two, you will be having nerve plexus. So as these nerve plexus are present in the muscle coat, it is called as myendric nerve plexus. Myendric nerve plexus. So already you have studied in physiology. Now one more thing, in stomach you will be having inner to these muscularis externa, inner circular and outer longitudinal layer, you can make out another oblique muscle layer. So the cut section of oblique muscle layer you can make out only in case of stomach. So inner oblique, middle circular and outer longitudinal. If you come out, you will be having serosa. So here the serosa is the peritoneum lining the stomach. It is a thin layer of peritoneum you can make out. There the lining epithelium is simple squamous epithelium. Already you have studied the simple squamous epithelium lining the your serous membrane are called as mesothelium. So these are the features of your stomach fundus. So identification, first you have to see for all the four layers: mucosa, submucosa, muscularis externa and serosa. So four layers are there. Next, how to differentiate it from esophagus. So there the lining epithelium is stratified squamous non-keratinized epithelium. But here you are able to make out simple columnar epithelium. So next what I have to see this beaded appearance due to the parietal cells. Clearly you can make out in this slide. So they are the large cells eosin stained. So that gives that beaded appearance. Okay. So next if you go down you can make out that hematoxylin stained chief cells. So if you see this beaded appearance with all the four layers of GIT, that is your fundus of the stomach. Okay, so this is low power. In this only you are going to see this light. So this is a high power. I projected this to demonstrate these parietal cells. So next slide what you are going to see is the pylorus of the stomach. So pylorus of the stomach means once again you have to see for all the four layers. So definitely you will be having all the four layers: mucosa, then your submucosa. Next you will be having muscularis externa and your serous layers. So but in this slide we have only up to submucosa. So this entire thing is mucosa. So this is submucosa. So remaining layers are not given in this picture, but you can make out all the four layers there. Now you see this. So this is the mucosa. So to identify this as your stomach, see for gastric pit is there. You are able to make out that gastric pits. So what is the difference between the gastric pit of your fundus and pylorus? So here at the level of pylorus, you will, that gastric pit will be very deep. So the gastric pit will be deep. So deep to that gastric pit only we have the glands. So here we have that pyloric glands. So these are all pyloric glands. As that gastric pit is very deep, the pyloric glands are short. So the glands are short. So in the next slide I will compare both. I will show that both the slides of fundus and pylorus how to differentiate. So the first point to differentiate is gastric pit, that is very deep. Next, if you see the gland, so these are all called as pyloric gland. These glands are mainly made up of mucus secreting cells only. Mucus secreting cells, similar to that mucus net cells. In the all these acini, you can make out only that mucus cells. So now this is your epithelium. This entire part is lamina propria. Now this is your muscularis mucosa. So that means this is your mucosa. Next, submucosa, the same thing as I told already. You will be having moderately dense connective tissue with blood vessels, nerve plexus. So the nerve plexus already I told, mucinous nerve plexus. You will be having blood vessels, lymphocytes. You will be having elastic fibers and collagen fibers. Next, outer to that, you will be having your muscle coat, that is muscularis externa, with inner circular and outer longitudinal muscle fibers you can make out. So in between that muscle fibers, you will be having nerve plexus called as your meandric nerve plexus. Next, outer to that you will be having serosa. So the all the features are same except that gastric pit and your glands are made up of only mucous cells. Now you see that two slides. So this is pylorus of the stomach. So this is fundus of the stomach. So here you see the gastric pit is shallow. So because of that the glands are larger. So entire two-thirds of lamina propria is occupied by the glands. So that's why the glands are larger here. But in case of pylorus, as that gastric pit is very deep, the glands are short. Okay, so this is the main differentiating point between the pylorus and fundus. Of course, here you can make out that parietal cells, prominent parietal cells will be there in case of fundus, but here you will be having only light stained your mucus secreting cells only you will be seeing in case of your pylorus. So that's what you have to differentiate fundus and pylorus. So now I have told about esophagus, your pylorus of the stomach and fundus of the stomach. So I told how to identify these slides. What are all the identification points you have to write? So any GIT slide you are getting means basic 4 layers. Next, what are the features of mucosa? What are the features of submucosa? So in case of esophagus, your epithelium is non-keratinized stratified squamous epithelium. In the submucosa, we have mucus secreting acini. But in case of stomach fundus, mainly the parietal cells, presence of parietal cells and a shallow gastric pit. In case of pylorus, a deep gastric pit plus presence of your pyloric glands. They are mainly made up of mucus cells. So with these ideas, please see the slides of esophagus, stomach, fundus and pylorus. Get that mental picture. Then you read the whatever points given in the record. Then you draw the diagram. Then only it will be easy for you to remember the slides. Easy for the discussion also. So thank you students. So all these images are taken from D5 Atlas. You can go through it.